The overall goal of this procedure is to isolate morphology-specific reagents against TAR DNA-binding protein 43 variants present in ALS, utilizing a two-phase AFM-based radiopanning technology. This is accomplished by first subjecting a diverse phage library to multiple rounds of negative panning against off-target antigens using immunotubes. Negative panning is performed sequentially against bovine serum albumin, aggregated alpha-synuclein, and healthy human brain tissue. The second step is to complete AFM verification after each set of negative panning to ensure successful removal of all unwanted phages. In this negative panning example using aggregated alpha-synuclein, phages are visible after one round, but are absent after the 10th round. Next, the remaining negative panning rounds are performed on mica due to limited sample availability. Phages reactive to immunoprecipitated TDP-43 from healthy and from frontotemporal dementia, or FTD, tissue are removed sequentially. The final step is to isolate the phages that bind to the ALS immunoprecipitated TDP-43 using mica. Bound phages are eluted using trypsin, triethylamine, and TG1 cells. Ultimately, the specificity of these potential ALS clones is verified using indirect phage ELISAs and dot blots. Homogenized ALS, FTD, and healthy human brain tissues are immobilized in both assays. Greater reactivity to ALS tissue is usually detected compared to the other tested groups.